Hi everyone! Today we are going to provide an overview of a plant pathogen that is responsible for countless devastating plant deaths across the globe, called downy mildew. I'm Annika.
I'm Arman.
I'm Allyson, and I'll be starting off this video talking about the developmental process of downy mildew.
First off, what is downy mildew? Downy mildew is a disease caused by pathogens from several fungus-like species known as obligate oomocetes. Oomocetes being a category of unicellular protists, and obligate meaning they're unable to live without a host plant. Though they resemble fungi, molecular studies and morphological differences put them in the kingdom Chromista. How to spot them? Downy mildew first manifests as circular yellow spots, also known as oil spots due to their oil-like appearance, 5 to 7 days after infection. As the disease progresses, the colors may turn brown or black and the leaves become neurotic and curl upward. And under warm human conditions, the fluffy white fungal growth from which they get their name will appear on the abaxial or bottom side of their leaves. The disease can also impact the plant's stems, flowers, and fruits as well. Now, how do they develop? Being oomocetes, they can reproduce both sexually and asexually, and their infection occurs in 2 infection cycles: primary infection spread from soil to foliage, and secondary spread from leaf to leaf. Their life cycle begins with sexually produced oospores, which can lie dormant in little dead leaf piles until their preferred environmental conditions. It's very common for species in many places to overwinter and reappear once spring arrives with new plant growth. Cool, wet conditions present the perfect environment for downy mildew to develop, and the oospore can begin to germinate. I now pass the mic to my groupmate, Annika.
Now that we have an idea of what downy mildew is and how it develops within various plant species, we can discuss the harmful effects of downy mildew within plant biology. Downy mildew pathogens, or DMPs, are usually categorized based on the species of plant that they infect. The most commonly seen form of this pathogen is Pseudoperonospora cubensis, which affects gourds, such as melon, squash, and cucumber. In order to gain a greater understanding of the varieties of DMPs, it is important to first understand where these species lie in the tree of life. According to genomic sequencing, researchers have found that the most recent common ancestor of oomycetes lies very deep within this phylogenetic tree, which serves to show that this is a highly persistent strain of pathogens that have evolved since then through their ability to reproduce. In order for this reproduction and dispersal to take place, however, peronospora require living host tissue, ultimately resulting in the death of localized areas of living tissue, and in some cases, the entire host plant itself. This ability for downy mildew to progress within plant tissue in such a short period of time is what makes DMPs such a big issue in plant development. One DMP that we can focus on is Pseudoperonospora humuli, which impacts hop. This particular DMP can cause entire crop loss from the infection of the cone-bearing branches. These infected shoots are called spikes due to their resemblance to a wheat spike and result in a dark brown discoloration. Some hop are more susceptible to the crown rot phase, causing the plant to die even without the formation of shoots. Another important DMP is basal downy mildew. Like hop, basil also lacks the genetic resistance needed to combat this disease. This is mainly due to the wide differences in genome makeup between basil species, oftentimes creating a barrier for sexual reproduction, and thus no resistance gene. In the United States, there have been resurgences of the disease observed, but it has been controlled for the most part thanks to the development of new resistant varieties and fungicides. An important aspect to consider when developing forms of resistance is understanding the exact mechanisms behind how downy mildew spreads.
Hi everyone, so now we're going to go over the mechanism for downy mildew, and for this presentation we're just going to focus on Plasmopara viticola. There are many other different strains of downy mildew, as you can see here, but the Plasmopara viticola was the one that I've seen that's most studied in the research, so we're going to focus on that specific mechanism, even though most downy mildew mechanisms are pretty similar. So it's going to start off with the formation of the oospores, and just to start, Plasmopara viticola is an oocyte that produces oospores, and it has two sex organs, Antheridium and Haghighinia. And what's going to happen is fertilization needs to happen, where the Antheridium will have a lot of nuclei that are in the Antheridium, and one nucleus needs to go through the fertilization tube and eventually go near the nucleus of the oospore to form a diploid oospore. And you're probably going to wonder what happens to the rest of the nuclei. Well, they just go into the periplasmic space to eventually degenerate. And one cool example is here, where you can see these stains of nuclei in the Antheridia and Haghighinia. Now that the oospores are fertilized, it becomes a waiting game. The oospore will stay dormant throughout the winter, and in the spring, when conditions are most optimal, the Plasmopara viticola will be able to use the water droplets in the humid weather and rain to transfer onto the leaves of nearby plants. So, now what is the optimal time for the oospore to germinate? It will germinate, and nuclei from the oospore will migrate along a germ tube that will be growing when it's time for germination. And once again, the germ tube is basically a structure that forms during oospore germination. So right here you have your oospore in this image right here, and then you also have some nuclei, as you can see, like the stain, and then you also have your germ tube right there, and basically those nuclei are going to head up the germ tube. And then on the right you have something different, where you have your germ sporangium actually form right here with nuclei in it, and you'll notice that there's no more nuclei in the oospore or the tube. And this germ sporangium is important because it will then release the uninucleated zoospores, which are the source of the primary inoculum. And a primary inoculum is basically the first original infection in spring after the oospore has been dormant the entire winter. Now it's time for the zoospores to do their thing. So once the zoospores are introduced to an organism, which is the leaf in this example, the zoospores will form their own germ tube and will grow into the stomata, and they'll have a vesicle in the stomata that'll give rise to haustoria. So in the first image, you can see your germ tube right there, and then there's your little vesicle. And this is what a haustoria would look like. So basically, they're going to be penetrating the cell wall, making room for their self, and basically haustoria are root-like structures that penetrate tissue of the host organism, which I've hinted at before. And now, once it's in the leaf, the symptoms are going to start beginning to appear. So growth of the Plasmopara viticola is going to occur, but it's going to get enclosed by the veins of the leaf, as you can see here. And then you can see inside this yellow stuff is the hyphae, and which are the filaments that make up the mycelium of the fungi, which is the Plasmopara viticoli. And after a couple of days, you'll notice, since they're trapped in specific spots because of the veins of the leaf, you'll get these typical yellow spots on the leaf, as you can see there. And there's also another symptom of downy mildew, which is common, that you'll see right here. And that happens when it's very humid, and the hyphae coils will grow under the stomata and give rise to sporangiophores, which will come out of the stomata, giving rise to sporangiophores that will grow out of the stomata. And basically the sporangiophore is a special type of hyphae which will give rise to sporangium at the end, which will make spores, allowing for the expansion of the Plasmopara viticola to other parts of the plant and maybe adjacent plants as well. And this here is known really as the downy mildew appearance looking thing, as well as this yellow spots being a pretty common phenotype / symptom of the downy mildew. So that's basically the whole mechanism. Hopefully you now have a more complete understanding of what downy mildew is, how it propagates, and how it affects plant life, and the mechanisms behind its development.
Thank you for listening.
And we hope you have a plentiful day.